Yeah, thank you very much for having me today. Hello, everyone. And I will try to answer this question, but actually we don't have answer yet. Food variation and susceptibility to naturals be linked to microbial communities. As you already could see from the previous talks, some microbes probably can influence natural resistance, and sometimes we really see that trees which are located very close to each other, like literally few meters away in the city, one tree is infected, another tree is healthy. For Pukutukawa, it's quite common that some individual trees are infected in the city center and in city parks, in urban parks. They are heavily infected. Sometimes you can see defoliation, like on the right side. Sometimes you can see only origina, only pustules, and sometimes there are only hypersensitive response, no origina present. So the initial question was, is it random or something is happening with the trees? If you see these very infected, heavily infected trees, they will pass infection to other trees nearby or not. We employed during several summers, several summer students to check this susceptibility. And initially we found 100 trees in the city center, right around the University of Auckland and in several parks, in Albert Park, or Devonport. And we checked exactly what is for each tree percentage of tree shaded, because as you probably know, when trees are shaded, probably there are high humidity, maybe better conditions for naturals. Also, distance to the nearest road, because we thought maybe from the road can be some, for example, pollution levels, so they can affect trees are more stressed, as a result they are more susceptible to relapse. Then stop growth flushes, because as we know, sometimes when we have new flush, and a lot of this new flush on the tree, we see high infection rate. Also, we checked for the distance to the nearest Myrtle, and after all these reports, we also counted basically how many origina are present on the tree. So we were just walking during 2 minutes around the tree and counting all the origina which we could see on the list. What we found, and again, initially it was 100 trees, but because some trees were later damaged, some trees were removed, and some data was inconsistent, in my opinion, not in our opinion. So it reduced to 86 trees, individual trees, to 3 consecutive summers. For the first summer, 2022-23, it had very high infection rate, because that summer was wet, warm, the best summer for Myrtle rust. Not so good for our Natasha, because many Pukutukawa were heavily infected, and we thought few summers like this, and there will be just no Pukutukawa, because all new growth in some trees was infected. But next 2 summers were much better for Pukutukawa, less better for Myrtle rust. You can see that between the first summer season and the next, there is significant difference in infection rate. And infection rate is exactly how many origina were counted just in 2 minutes. For the first summer, we counted up to 150 origina in 2 minutes. So you just counting on the list, and you should just think that it's actually not origina itself, but leaves. I mean, 150 infected leaves on the tree in 2 minutes. If you continue to count, sometimes everything will yellow. When we try to understand which factors can influence this infection rate, Actually, you can see regression analysis did not show significant difference in shaded, not shaded trees. There was very weak positive relationship between mean flash lengths for some trees. So when trees they had longer flashes, a lot of new foliage, it was positive correlation with nodal rust infection. Also was negative correlation with relationship with distance to nearest road, and very weak relationship between distance to nearest infection. So we were checking individual trees during 3 seasons, and we still could see that some trees were not heavily infected, being close to heavily infected. And why? It was a big question because some of them were probably hybrids with Kermodeckensis. And some observations suggest that hybrids with Kermodeckensis, they are more susceptible to Metrosideros. But other trees, they were clean Puhutukawa. And I even did a small blind test. I sent samples from heavily infected tree and not so heavily infected, which were very close to each other, to a botanist, well-known, Peter Delay. And he said, oh, this, which is number 1, it is probably hybrid with Kermodeckensis. This, which is number 2, it is probably clean Puhutukawa. But he did not know what exactly was infected and not infected. And after several samples like this, we just thought maybe Kermodeckensis somehow influenced when it's a hybrid with Metrosideros excelsa. However, for clean Metrosideros excelsa, we also found that some trees were more infected than others. And we tried next to do bootstrap analysis. So we just measured mean absolute difference in infection rate between the first and third summers. And after we compared this mean absolute difference with all our trees, so what we did, a random tree from the first year was compared to a random tree from the third year, and this algorithm was repeated 10,000 times. What we found that mean absolute difference in infection rates for some individual trees was much lower than we would expect that infection was randomly distributed. So probably some individual trees, they have some resistance. What is resistance? It can be genetic resistance. It can be probably used by microorganisms, and it probably can pass to next generations. And here a big question, if we speak about genetic resistance, how it is also linked to microbial resistance, because some microbes, when they are present inside of the tree, they produce plant hormones, many bacteria, they produce plant hormones, fungi produce plant hormones. Also, they can interact with microbes directly, for example, not rust-derived, and there are already tophytic communities which are present inside of the tree, which just don't want to be, let's say, in the competition with this new arrival. So many bacteria and fungi, they can directly suppress not rust. And some of them can pass vertically to next generation. So next part of this research was to check what's exactly going on in healthy trees. We collected flower buds, seed capsules from 6 locations around Auckland. You can see it was 3 regional parks and also in the city center areas. In each place, we just randomly chose 5 healthy looking trees. From these healthy looking trees, we collected flower buds and seed capsules. Seed capsules were collected before they were opening, so still closed. Seeds were intact inside. Seeds were aseptically extracted, so we surface sterilized flower buds, we surface sterilized seeds, and seed capsules, surface-derived seed capsules, and extracted seeds. After that, bacteria and fungi were isolated from seeds and flower buds. Also, seedlings we obtained from seeds, so seeds that were germinated. And also, we extracted DNA for further analysis of bacterial communities and fungal communities. Initially, when we obtained cultivation results, it's just an example. There are more, actually, it's already published. But I thought that it is a big cultivation bias because here on the graph you can see actually individual trees and the relative abundance of most frequently isolated actually 7 species, 7 genera, and the others they were marked just like others. We can see that individual trees they accumulated quite different fungal communities. And I thought maybe it's just cultivation bias, nothing more. But later, when we checked metabarcoded results, here again, only an example for when there are five trees, and on the left, bacterial communities in flower buds, seedlings. On the right, fungal communities in flower buds, seedlings. You can see with different colors, different clusters of bacteria and fungi. And again, we see a difference in microbial communities among individual trees, which was quite unexpected because we thought in each location, microbial communities should be more or less similar. But there was one issue with all these locations. Many trees are planted. And the source of the trees, provenance of the trees, can be very different. So in Wenderholm, some trees can be planted from as far away as from Taranaki region, initial progeny, or from far north. And we already did some preliminary analysis of genetic diversity of these trees, and we found that, yeah, actually, these trees, they have different genetic, and they are coming from different sources. So it is probably why they contain different microbial communities, because they could originally accumulate different microbial compositions in the initial source, and after that, when they came as seedlings, they already had something inside. When we speak about culture-based results, we isolated 17 species of bacteria and 25 species of fungi. Some of them were beneficial for seedling survival, some of them are detrimental, and we also had actually several seed-borne pathogens in seeds. Here, this is on the right, it's a very good bacterium which accidentally formed this tree. I was not painting the tree, it was just accidentally. Anyway, when we germinated seeds, we found that for individual seed germination rate was very different, actually also. Normally, healthy pocotocolor seeds, they should germinate around 80% and 100% germination. But some trees, they did contain seed-borne pathogens, and actually they all failed just after germination. You could see that there are mold around, and this mold was Anophococcum. Anophococcum species, they usually not vertically transmitted, but with exactly this tree was very interesting that it did not contain core bacterial community. The two general core bacterium were missing in seeds. And in this case, I can speculate, okay, Wenderholm 1, all seeds are gone because core bacterial community, few genera were missing completely, and missing according to metabarcoding analysis and also according to cultivation analysis. So it's, yes, but we actually need more data to say that it is a case for the trees, because it's only 3 out of 30 trees. We saw that missing bacterium was also somehow correlated to failing for of the seedlings. When we checked how bacteria can suppress actually natural germination on media, 14 bacterial isolates were chosen based on literature records for exactly this bacteria, which will suppress in other rust species. So, for example, it can be wheat rust, it can be peanut rust, coffee rust, and you can see in literature records that these bacterial taxa, they suppress other rust species. So we found that one Bacillus isolate was effectively suppressing natural germination. So you can see original spores germination, lower part it is bacterial colony and not so many spores could germinate. And upper part you can see all the germination tubes appeared. So we can speculate again that if you apply exactly these bacteria on the surface of the leaves, probably germination rate will be lower for Myrtle rust. But unfortunately, I did not have time to do this because all plants came to the end. Anyway, this inoculation experiment demonstrated that if we initially sterilize seeds with different solutions of antibiotics and fungicides, it will affect seedling development, but still we cannot sterilize seeds so that there are no bacteria, no fungi present. Even with systemic fungicides, even with antibiotics, we still cannot eliminate microbial communities. And that's why I'm asking about sterilization protocols, because as far as I know, many tissue cultures, they contain a lot of beneficial microbes inside. And we actually should preserve these beneficial microbes, because you can see on the lower part of this picture, when we inoculated seeds after the sterilization protocols, with on the left we see penicillium, in the middle is Fukuria present, and on the right Pantoia. You can see that seedlings developed better root architecture, a lot of lateral roots. Also, it's quite interesting that this penicillium, somehow all the seedlings were only a single stemate. There are no branches. But with bacteria, sometimes you can see multi-branched seedlings, and some bacteria, they produce seedlings which are mostly with many branches, many roots, and now when you see all the trees around, some of them with a lot of branches and a lot of additional roots, and some of them just mostly single stem. And you think what's going on inside of these trees from the beginning. Maybe different microbial communities produce different phenotypes, because they can be viewed as extended phenotype. All these bacteria and fungi, they produce hormones, they produce other substances for plant growth, and yeah, it's all the seeds derived from only one tree. Of course, we can think about diverse genetic diversity of seeds from one tree, because of course they were pollinated probably from different trees. But still, you can see quite a big significant difference in terms of seedling appearance, and I can assure you that there are also significant difference in shoot weight, in dimensions, all this. So we checked it. We'll use statistical analysis. I just don't have time. Okay. If someone is really interested, I can share all these results, and it's already under review for publication. In summary, unfortunately, I still cannot say that microbial communities are literally linked to resistance of Fukutokawa, but we can see that individual trees demonstrate different rates of susceptibility to Myrtle rust. And during 3 consecutive summers, we could see that these individual trees, they are not infected, or infected only mildly, being close to heavily infected. Next, we can see that seedlings derived from each tree, they retain specific microbial communities. So if you think about individual trees and they produce seedlings, and these seedlings, they markedly different microbial communities derived from parental trees. You can think about susceptibility of these seedlings in the future to pathogens, including nodulus. So if experiments demonstrate that microbial communities can influence germination and plant growth susceptibility to pathogens, including nodulus, we should think how to apply this. And there are a few very big and sometimes controversial questions. Is it possible to transplant potentially beneficial microorganisms from one potato cover to another? Because we should consider papa, we should consider progeny of the disease, we should consider different ethical and other approaches. Is it possible to transplant microbial communities which are beneficial from Pokutukawa to other species of mutation? For example, we have Silicium, and we know that actually Silicium or Rama locomotus, they will never meet Pokutukawa in normal conditions just because they're just plants of different habitats. Pokutukawa is a coastal tree, and Rama is a forest tree, bush. And the last question, and it's very also a big controversial question, is it possible to use some microbial communities derived from wild plants, native plants, maybe for speeding up growth and production of wheat, oats, and other agricultural crops? Because actually what we are doing, we are a little bit tinkering with evolution. In natural conditions, many plants, they can acquire microbial communities from unrelated plants because of wind, pollinators, and other vectors. But it is quite long process. And we should think, can we work a little bit to speed up this process or not? So that's all from me for today. This work was cumulative report of many people, my great supervisors, Bruce Burns, Mahadjabin Padanski, and also Haley Ridgway. Renee Jorgensen was our great manager for Metrosideros from the beginning. Also, 3 summer students during 3 consecutive summers, we were collecting all this data from 86 Pokutukawa trees. And yeah, I'm very grateful to all teams at Myrtle here in Auckland, in Lincoln, and wonderful team. Thank you very much.